Hey everyone, welcome to a high-speed explainer where we're basically turning Brenner's complex kidney physiology into a game show. Today, we are testing your knowledge on exactly how neurohumoral factors control salt balance in the nephron. So get ready to lock in your answers.
All right, here are the rules. You're the contestant today, and your job is to shout out whether the hormone we're talking about increases or decreases sodium absorption. Think you can handle it?
And just so we're all on the same page, our entire game board today is derived directly from a true masterpiece of medical literature, Figure 6.9 of Brenner's 11th edition. This figure brilliantly maps out the exact neurohumoral influences on sodium chloride absorption across the entire nephron. There's literally zero guesswork here. This is pure, grounded physiology.
Okay, let's jump straight into round one, the pro-absorption heavyweights.
We're bringing in the absolute heavyweights of the nephron right out of the gate here. We've got angiotensin II, aldosterone, and renal sympathetic nerves, which of course operate via norepinephrine. So here is your question. Do these three increase or decrease NaCl absorption? And which one dominates the proximal tubule? Lock in your answer right now. If you yelled increase, you absolutely nailed it. These hormones drive massive sodium retention. They're basically the body's ultimate defense system for maintaining extracellular fluid volume.
And what's super cool is how these heavyweights divide the labor beautifully across the nephron. Angiotensin II and norepinephrine, they massively drive proximal tubule absorption. Norepinephrine cranks up sympathetic tone, while angiotensin II operates on both luminal and basolateral receptors to just turbocharge that sodium-hydrogen exchange. Meanwhile, aldosterone completely bypasses those proximal segments. It handles the distal tubule and collecting duct, rapidly upregulating apical ENaC channels to lock down that final crucial percentage of sodium. It's an incredibly efficient tag team.
Round 2, the natriuretic counterattack. Now we're looking at atrial natriuretic peptide, dopamine, and prostaglandin E2. Do these three increase or decrease NaCl absorption? And what's their ultimate goal? Tell me now. Boom, you got it. These are the absolute decreasers. They are slamming the brakes and driving sodium absorption straight down. The crucial takeaway here is that these are the body's volume depleters. They work dynamically to excrete sodium, which is a process called natriuresis, by actively inhibiting transporters. It's fascinating because dopamine is actually synthesized locally right in the proximal tubule from filtered L-DOPA. Then ANP comes in, sensitizes the tubule to that dopamine, and together they dramatically inhibit proximal absorption. Further downstream, PGE2 puts a massive block on the thick ascending limb. This ensures that sodium stays in the lumen and is flushed right out of the body. All right, round 3, the tricky modulators. Let's look at arginine vasopressin, also known as AVP or ADH. We all know AVP acts to absorb free water in the collecting duct, right? But here's the trick question for you. Does it increase or decrease NaCl absorption specifically in the thick ascending limb? What do you think? Quiz pause, 3 seconds. Surprise! It is a massive increase. Yeah, AVP doesn't just manage water. It actually stimulates the apical NKCC2 transporter in the thick ascending limb. It rapidly triggers the exocytosis of these NKCC2 proteins straight from subapical vesicles directly into the plasma membrane. This completely ramps up sodium chloride absorption, which brilliantly helps build that vital medullary concentration gradient. And think about it, without this gradient, AVP literally wouldn't be able to pull water out of the collecting duct later on. It sets up its own success.
Final summary, the nephron tug-of-war.
You've survived the gauntlet. Now, let's lock this knowledge in permanently. Picture this whole system as a massive physiological tug-of-war. On the left side, we have our increasers pulling hard, working tirelessly to retain volume. And on the right side, the decreasers are pulling back with equal ferocity, trying to excrete that volume. Looking at the left side of our rope, these increasers absolutely dominate their specific anatomical territories. Angiotensin II and norepinephrine completely secure the proximal tubule. Then AVP handles the thick ascending limb, maximizing energy efficiency. And finally, aldosterone locks down the loop and distal segments, making sure very little sodium escapes. But fighting right back on the other side, the body has this robust defense against volume overload. Dopamine and AMP inhibit proximal absorption to create a powerful natriuresis, while PGE2 puts the brakes directly on the thick ascending limb. Together, they effectively counteract all those heavy weights on the left. So, considering this delicate, highly regulated balance, which side of this neurohumoral rope do you think completely takes over when a patient's blood pressure drops critically low? Think about who wins that ultimate physiological tug of war to literally save the patient's life. Hold on to that thought, and we'll see you in the next explainer.